Okay, everyone, I just wanted to talk a little bit about energetics and enzymatic reactions. I know we skipped over this in our, we discussed enzyme kinetics last week. So I just want to throw this in to fill this out and round it out for you guys. So I just want to remind you all that, you guys remember the delta G equation here, the standard free energy change equation from chemistry, I'm sure, right? Delta G is a Gibbs free energy. It's the measure of work, of energy available to do work in a system. Delta H is enthalpy, basically the measure of heat in a system or the total energy in a system. And then delta S is the change in entropy in a system. And those enthalpy, change in enthalpy, delta, which is change, the symbol used for change, and the change in enthalpy minus a change in entropy, depending upon temperature, T, right, will determine the free energy in a reaction. And usually reactions are considered to be spontaneous or can run without energy input if this delta G value is negative, right? So the delta G is usually measured in reactions that we're concerned about in terms of the free energy state of the final state of the reaction minus the energy state in the initial state of the reaction. That's the delta G value here, right? So if the initial state of energy of the reaction is higher, right, than the final state, then the delta G value here is negative. And that's an indication of a reaction that will run spontaneously and release energy in as it runs, right? So a higher energy at the beginning state can be illustrated in a couple different ways. We're really talking about potential energy being converted into some other form of energy, right? In this case, so say a guy on a diving board here, right? He has potential energy, which he because he's higher on the higher up from the surface of the lake that he's going to dive into. He's has more potential energy, right? Against measured as against basically gravitational forces, essentially, right? He has a greater work capacity, especially if he converts his body into kinetic energy as a result of dying off the board and going closer to the gravitational source that he is attached to, right? So he's essentially a less stable state. So his energy state's higher, right? And then when he dives, as you would here, right, he converts some of that potential energy into kinetic energy. That's movement or motion, right? And gravity's powering that, right? And so as he dives and changes energy, his body into kinetic energy from a standstill, right, he actually is decreasing the free energy of the system. He's going towards a stable state as he drops in altitude, right? And as he drops, of course, his body can do work because it's actually moving and has kinetic motion and can actually power work, which is what delta G is all about, is measuring work, capability of doing work. The end state is when he reaches the water and collides with the water and then is bobbing there in the water, right? He has less free energy than what he started with. He's more stable and he has less work capacity. Now, where is that energy gone if he hasn't actually used the energy to do any work just by falling through the air? He's not attached to any ropes or pulleys or anything and doing any kind of particular work, right? Where's that energy going when he hits the water? Well, he's heating up the water when he goes in. So when he dives in, he pushes the water molecules out of the way, breaks all those hydrogen bonds, right? And converts his kinetic energy into heat, which is now resident in the lake. And that should be measurable, actually, whenever someone dives into water like this. Okay, now, other examples of potential energy sources that can be utilized and when they're allowed to go to a lower energy state is, for example, diffusion, where you have a lot of molecules at one side of a dish of water, let's say. They have a high energy state, they have low entropy here, right? This part of the equation is lower, right? And then when these molecules are allowed to diffuse freely, let's say they had a membrane around, that membrane's removed, or holes are developed in that membrane, and these things can diffuse. They fuse until they are equally distributed throughout the dish of water, right? And that's maximum entropy. So the entropy actually increases here, and this value then goes very high, right? And since it's negative in the this part of the equation, delta G value goes down, right? So this is an energy high potential energy, lower potential energy, right? And so the change, when this happens, is going to do work. And actually cells use this all the time to generate energy to be used for doing work. This is basically how ATP is made in a cell. This is a high energy, high concentration of protons on one side of a membrane, right? Lower concentration of protons on the other side of the membrane. And when those protons move down their concentration gradient through an ATP synthase, that powers ATP synthase to make ATP and results in protons being, the concentration of protons across the membrane being equalized, and the protons actually reaching maximum entropy state, right? And that results in ATP synthesis, which can then be used to do work. And then there's chemical reactions that have chemical energy, right? So here's something like glucose, right? Which when combined with oxygen, can be broken down into smaller molecules, right? More entropy in this state than in this state, right? More disorder, of course, and release of a lot of free energy, and again, an increase in energy state of the reaction of the system. So that are three ways of energy stored in potential energy, which can be used to convert into work of some sort. Okay. When we're talking about enzymatic reactions, we can measure them in a free energy state, or any chemical reaction, actually, in the free energy state over time, or the progress of the reaction on the x-axis here, free energy state on the y-axis. And what you'll know is that certain types of reactions will tend to have a high free energy state to start with, right? High delta G value. And then when the reactions are allowed to proceed, energy is released and the products have a lower energy state, right? So that's a negative delta G value. That's called an exergonic reaction, a good word to know. Energy is released. This energy, if it's captured in some way, can do work. This measure of that energy is the difference between the energy state of the reactants and the energy state of the products, right? And that value is less than 0. And so it's a spontaneous reaction, it's exergonic, and energy is released. You can have the opposite happening too, right? So you can have reactions where you have reactants with a low energy state, and then products with a higher energy state, right? And these are going to require energy input. So we have energy being put into these reactions, and that's going to be what's called an endergonic reaction, energy required, and the amount of energy is going to be measured by the difference between the reactants and the products. In this case, it's going to be positive. So that's an endergonic reaction, and this is going to require energy input to make happen. All right, those are the two main ways we're going to see enzymatic reactions run in cells. This is going to be usually catabolic reactions in cells, reactions that release free energy for use in doing work, and anabolic reactions are reactions which need energy to carry out, and those are usually reactions that are going to be making things, synthesizing things. This is by building blocks are needed for the cell, for example, where you're taking those building blocks and breaking them down in exergonic reactions. I'm sorry. Okay. So what's the main energy currency of the cell, and how's that cell going to be storing energy mainly? Well, ATP is the big way. Here it is. You guys should be familiar with this. Adenine base, ribose, 3 phosphates. And where's the energy in this system? Right in these phosphates. All right. These phosphates are very, very energetic, and you can see why. We have a lot of negative charges here, all in close array. These are right next to each other and very, very close. These negative charges are repelling and do not like being here. And so this is very unstable. This is prone to hydrolysis at the phosphate here, and at these oxygen diester bonds between these phosphates. And so these tend to hydrolyze easily. And when they hydrolyze, these phosphates run off and increase entropy of the system. And of course, maybe some kinetic or and heat energy is released as a result of this. And the energy state of the system warms up. It's an exergonic reaction, right? Now, most cells and most reactions in a cell do not just lose phosphate, and phosphate goes off and somehow magically makes reactions happen. This is actually often done in such a way that you take this phosphate, add it to another molecule, increase its energy state, and then it allows it to actually carry out a reaction that it couldn't do before. So that's what ATP is often involved in. We'll get into that in a second. Okay, so hydrolysis ATP results in the release of energy, which can be used to do work. Here we have our ATP schematized, right? And you remove phosphate. This is called phosphate, inorganic phosphate, or IP, right? I stands for inorganic. And when you hydrolyze this reaction, you get energy here, right? But of course, in a cell, this is not just as energy just floating around to be used in reaction in some sort of magical way, right? This is actually energy that's released only because this phosphate goes somewhere and does some sort of operation that allows a molecule to do what it couldn't do before. Usually it's an addition of a phosphate to something that increases its energy state, and then it can now carry out a reaction with a favorable thermodynamic property. Okay, so here's an example, right? If we are making the amino acid glutamine from glutamic acid has a carboxylic acid group on the R chain of the amino acid, right? And to make glutamine, you need to add ammonia to that. So if we just link glutamic acid with ammonia, right, through just normal chemical reaction to make glutamine, this has a delta G value of plus 3.4 kcals per mole, right? So this is an endergonic reaction, anabolic, right? And that is not spontaneous. This will not happen easily. You can make it happen if you fiddle around with the equilibrium of the reaction some, but basically you change the reactants, like you increase the reactant concentration to a very high level, and very little bit of this, you can make this reaction happen to some extent, right? Without putting energy into it, but to a small extent. And towards delta G is going to reflect equilibrium equilibrium state of these reactions. But what the cells figured out to do is actually what you can do is convert this into an exergonic reaction by taking ATP and hydrolyzing it, and in the process coupling that energy release from the phosphate being released in ATP here to this reaction. Essentially what you're doing is you're adding this phosphate to, think glutamic acid here, right? Increasing its energy state and making the ammonia more likely to react with it. And then the phosphate leaves as a leaving group, and this now makes this. And when you do this, hydrolysis of ATP releases energy to the tune of minus 7.3 kcal per mole. And the formation of glutamine from glutamic acid and ammonia has a delta G value plus 3.4 kcal per mole. In these reactions, you can simply just add these values together. So if you couple this reaction, ATP hydrolysis, to this reaction, then you just add the values together. And the overall reaction is going to now be very exergonic, minus 3.9 kcal per mole. And now this is spontaneous. This reaction will run on its own, right, and work very well. And so ATP is very useful in the cell for making things like this happen. Okay, so overall then, the cell is taking energy from catabolism, energy yielding processes, metabolism of glucose, for example, through cell respiration to make ATP. That ATP then is used to do work or anabolic reactions, right, to produce more complex molecules, for example, or for doing actual cellular work, moving things around or whatever it might be. Hydrolyzing ATP in the process, and then catabolic reactions continue on to re-phosphorylate ADP with phosphate, and you go around a circle like this. And in the process, you are breaking things down and releasing energy, and then building things or doing work and gaining energy. All right. Okay, so what's examples here? Motor proteins moving vesicles down cytoskeletal tubules, right, require ATP hydrolysis to attach a phosphate to the motor protein that helps power the motor protein, make it change conformation so that it actually walks down this microtubule, right? And ATPs need to do that. The sodium phosphate, sodium potassium pump, we'll talk about in next week, actually later in the week, allows active transport, which is allowing movement of ions, sodium, potassium against their concentration gradient. And to do that, you need ATP, and phosphorylation of the molecule powers this, right? And then, of course, here we have this example I just showed you, where we have glutamic acid being have an ammonia added to it, and the way you do it is by adding a phosphate from ATP, and then the ammonia is more likely to react in the enzyme that carries this reaction out, and you get glutamine and phosphate, and that can go on to make ATP again through catabolic reactions, right? Okay, so that brings us to enzymes and enzyme reactions that we went over with last week. So I just want to make sure you make that connection, right? Now, usually in reactions that we're talking about that are exergonic, we're going to have reactants at a higher energy state versus products are going to be lower energy state, negative delta G. Spontaneous reactions, this is spontaneous exergonic reaction, right? You can also have it this way, right? I added this since last time we talked. Here is an exergonic reaction, minus delta G, right? But an endergonic reaction can also be happening. This could be catalyzed by enzymes too, right? Where the enzyme still lowers the energy of activation, but you're going from a low reactant energy state to a high energy state of products. Delta G value is positive here, right? And an enzyme would catalyze this, and you'd need some way of bringing energy into the system to make this happen. Okay, so that's enough of that. Hope you found that helpful.